Okay, we'll start. The first speaker this morning is Eran Segal. He's from the Weizmann Institute, Israel. He's going to talk about cracking the regulatory code.
I'm on, okay. All right. So good morning, and I'd like to thank the organizers for inviting me to speak at this very interesting workshop. So my lab is mainly focused on studying gene regulation at several levels, but mostly on the transcriptional level. And the long-term vision is to be able to look at a piece of regulatory DNA and just be able to read it and understand what type of transcriptional behavior it's actually encoding for, and hence cracking the regulatory code. And the title is not because I think we're there, we're actually very far from the solution, but just to outline the general vision of where we're headed. And if you think about it, it's actually it's a very hard problem to solve. It's like if somebody were to give you a book in a language that you don't understand and took away all the punctuation marks and didn't even tell you what the words are, what the sentences are, what even a word should look like, and so on. And so if you imagine that somebody did that, then it would probably look like trying to read something like this. And so that's essentially our challenge. And today I'd like to tell you about, nevertheless, despite the difficulty, some progress that I think we're making in this long-term goal. Okay, so transcriptional regulation, the process by which cell decides when and where to activate their various genes, important in nearly every biological process, from processes such as cell cycle that happen at the single cell level up to processes at the organismal level, like, for example, being able to activate specific genes at precise temporal and spatial locations along a developing embryo. And so the way this process works is every gene has two regions, coding regions coding for the protein product of the gene, and a control region that carries the instructions for when and where to activate the gene. And this control region contains several individual words that are recognized by designated proteins called transcription factors, and by the binding action of these factors to the DNA, they can activate appropriately the corresponding genes. And so what we have is a program in the DNA that encodes for a certain behavior. And so if you're a computer scientist, this has perfect analogy to computer programs, where you also have programs written by engineers that encode for specific behaviors. And so if you're a computer geek and into computer science, you probably played these games a lot, where somebody gives you a behavior and your goal is to design a program that will exactly achieve that type of behavior. And so why not strive for trying to do something similar with biological systems? Somebody one day may be able to give us a behavior, and we'll be able to write a program that will encode for exactly that behavior. Okay, but we're still very far from that because, as I mentioned, we really still don't understand how the basic components in the system interact. And so I mentioned factors binding to DNA, but in fact, the picture is much more complicated because you actually have a network within the cells whereby every transcription factor is responsible for controlling the activity of multiple genes, and conversely, every gene is controlled by multiple transcription factors, leading to these regulatory network structures that people have been trying to infer the structure from many different types of data. But what we're going to be talking about today, what I'll talk about today, and what our interest now is in trying to understand just how exactly are edges in general in these types of networks implemented within the cells. And despite a lot of study that has been going on in transcriptional regulation, actually really the very basic questions are still quite open and poorly understood. So, for example, transcription factors recognize specific words within DNA, but really we still don't know the binding specificity of transcription factors. Martha Bulick, who'll be talking here today, has been doing very beautiful work in advancing our understanding of the binding specificity, but largely this is still an open problem. If you continue on, you find in promoters that there's additional higher order structure like clustering of binding sites, both strong and weak binding sites and sites with different orientation, but exactly how this plays out in determining the regulation is not known. Then the exact mechanisms of how factors activate or repress are not understood, like how important is cooperativity between transcription factors, and does repression occur by a repressor occluding the activator from binding DNA, or perhaps by both activator and repressor binding, but repressor quenches the activity of the activator. DNA looping is known to play a role, but is perhaps one of the most poorly understood processes here. And of course, all this happens in the context of chromatin structure, so how does this figure into the game? And finally, even if we could understand all of this picture here, this would indicate that we understand the process of factors binding to DNA, but it would still leave open the question of how all these binding events then produce transcriptional behavior or function. And so our main goal in working on these problems is to see whether we can develop mechanistic quantitative models for describing these processes at the transcriptional level. And I'd like to tell you about two such models that we've developed for the system today. Before I do that, I'd like to just mention why it's good to stop and pause and think, why is it even a goal that we'd like to achieve? Why even develop models for the system? And there's at least three reasons that I would mention. First is maybe my belief is it's only when we get to having a model that's predictive of a system that we could really claim that we actually understand how the system is working. Then if you have a model that explains well the system, you can actually use it to distinguish between different mechanisms by seeing which type of model with which assumptions is actually is better at explaining your data. And finally, of course, if you have a model that you can learn from some data that you've measured, then you can use that model to predict behavior in situations that you have not observed before. Okay, so the two models that I want to tell you about today is one model for trying to understand transcriptional control during fly development from looking at how transcription factors are modeling the process of transcription factor binding to DNA, and if I'll have time, then on some new advances in models for modeling chromatin structure within eukaryotic cells. Okay, so for looking at transcriptional control, we chose the segmentation network of the fruit fly Drosophila melanogaster. This network is responsible for setting the anterior to posterior expression gradients of genes within the system that are then, which is important because that then determines the entire body plan of the fly. The system consists of a hierarchy of transcription factors, starting with maternal genes deposited at specific regions in the embryo. The maternal genes then regulate the broadly expressed gap genes, which then together with the maternal genes go on to regulate the 7-striped parallel genes. And the system has been extensively studied, which is why we chose this system, since many of the components have been identified, and we can now look at how they actually interact together to give specific functions. And from all the studies that have been done, we understand much about the structure of the regulation here, and the system is quite remarkable in the sense of its level of modularity. So you can take these genes that are expressed in 7 stripes, and you can look at the control region of a gene, and you could break it up into these different modular units called cis-regulatory modules, each consisting of hundreds to a few thousands of base pairs in length, and containing multiple binding sites for multiple transcription factors, and that somehow this combination then specifies 1 or 2 of these 7 stripes, and then you get the entire 7 stripes by having all these modules acting together to regulate the gene. Okay, so I mentioned many of the components in the system have been identified, and the question that has been less explored and was the subject of our studies is to try and see if we can understand how these DNA sequences are actually computing the expression patterns that they're each responsible for. So in other words, how is it that one DNA sequence looking at the set of inputs, gradients of inputs of transcription factors produces one type of pattern, whereas another DNA sequence present in the same cell, obviously looking at exactly the same inputs, produces an entirely different pattern of expression along the anterior to posterior axis. Okay, so to look at this, we developed a thermodynamic model for transcriptional control that has two main levels. At the first level, we try to model the process by which transcription factors bind to DNA, and at the second level, how these binding events translate into transcriptional outcome. And so the input is measurements that we have on the transcription factors in the system, including both their gradients of expression along the anterior to posterior axis and some information that we have on the binding specificities of these factors. And together with that, we have measurements of the expression patterns produced by different of these cis regulatory modules. And so the goal is to then be able to take a DNA sequence and then predict where along the anterior to posterior axis it will give rise to expression. Okay, so I want to give you the intuition and behind the two components of our model. Very briefly, the first part is modeling the process by which transcription factors bind DNA. We do this for each and one of the different cis regulatory modules that we have as input. And for each of those, we do it at every different spatial position along the anterior to posterior axis. So to understand the model, we need to understand how we do it for one cis regulatory module at one given position at the anterior posterior axis. So let's imagine that this is our cis regulatory module. And for now, let's make the assumption that somebody actually in advance told us where there's binding sites for these for transcription factors within this module. Okay, so if that were the case, you could imagine that one possible configuration of this cis regulatory module would be the one that's shown here, where the two different transcription factors, the three different molecules, would each be bound to the site that they recognize. Okay, so let's think about what would be the probability that we might find the system in this state. Well, you could think about the energetics of it, and if you make the assumption that, let's say, each of these binding events is independent, then all you have to do is understand the energetics behind one of these binding events. And behind one of these binding events, there's going to be two factors that are going to determine how likely is a binding event to occur. One of them is going to be the concentration of the factor that you look at. The higher the concentration, the more probable the factor will be bound actually at any position. And also, of course, the binding specificity that the factor has to this particular DNA sequence at this binding event that we're modeling. Okay, so under the independence assumption, you could then add up in energy space. You add up these energies, and you can get a statistical weight for the system to be in this particular configuration. Okay, of course, for these same organizations of binding sites, you can have different configurations of the DNA because only a subset of the molecules could be bound. But then for each of these, we just explained how you could model the statistical weight of that configuration. And if you now make the assumption that the system is under thermodynamic equilibrium, then you could actually compute exactly the probability that the system will be in any one of these given configurations by simply taking the statistical weight of the configuration and dividing that by the sum of the statistical weight of all the possible configurations. Okay? So then you can compute exactly the probability distribution that the system will be in each and one of these states. And so now the only thing that is left to do is, of course, nobody really tells us in advance where there's possible binding sites for transcription factors. And factors can actually, depending on, again, concentration and binding specificity, might be able to bind anywhere along this DNA sequence. And so really we have to scan over all possible positions for the binding of factors to this DNA sequence. This is a huge exponential space, but we have to sum over it if we don't want to threshold different sites. And the other thing that we'd like to take into account is steric hindrance between factors, because some configurations are just physically not realistic. Two factors cannot bind typically the same base pair of DNA. So you want to go over all possible configurations, but excluding configurations in which you have two factor molecules bound to the same base pair of the DNA sequence. So it turns out that even though the space is very large and exponential in the number of factors, you actually can solve this problem, this equilibrium problem, exactly using dynamic programming. And for each one of these configurations, you can in fact compute exactly the probability that the system will be in that configuration. Okay, so that's essentially the DNA binding component of the model. So the model explicitly integrates the concentration of factors and the binding specificity of factors, and explicitly models competition between factors as they bind DNA by modeling the steric hindrance effects between transcription factors. And by scanning the entire DNA sequence of a cis-regulatory module, we actually allow integration of both strong and weak binding sites for every given transcription factor. Okay, so the second component of the model then describes how these binding events translate into levels of expression. And so here the intuition is simple. If I look at a configuration and there's more activator molecules bound than repressor molecules, then that configuration will lead to transcription. And if there's more repressors, then that will lead to no transcription. And so formally, we capture this using the logistic function, where we sum up the different molecules that we have bound at each configuration. Each molecule has its own weight. Activators have positive weights, repressors have negative weights. And the sum of these will determine where along the x-axis you are, and then correspondingly where you are along the y-axis. And the logistic function also has the nice saturation properties, whereby after a certain level number of activator molecules, adding more activators will not increase transcription. You've already reached the maximal transcriptional rate. And conversely, after a certain level of repressor molecules, you're not producing any transcript, and adding more repressors will not change that situation. Okay, so finally we can explain how, for a given cis-regulatory module at a given position along the anterior-posterior axis, how we actually compute the expression that it gives. We simply sum over all possible configurations of molecules on the DNA. For each configuration, we compute its output expression, and then we simply sum this expression, sum it by the weight of being in each configuration, which we can compute exactly in the DNA binding component of the model. Okay, so that produces a predicted level of expression for a given module at a given position. And to get then predictions for the entire system, we simply run this for all modules across all different positions. Okay, so this model sounds complicated, but in fact, it actually has a very small number of parameters. In fact, only 2 parameters per each one of the 8 transcription factors that we have in the system. One parameter is for scaling its relative measured concentrations into absolute levels. And the second parameter is this parameter of the weight that translates the contribution of a bound molecule to the outcome expression. And so we take these 16 parameters and we fit these parameters such that we maximize the agreement between the predictions that we get for all cis-regulatory modules across all positions of the anterior-posterior axis. Again, we of course try to find, since these parameters they go with the factors, they should be universal for the system. So we try to find one set of parameters that jointly maximizes this agreement across all modules and all positions. Okay, so applying this model to the system, we could see that it actually did a pretty good job at being able to position the pattern at about the right place, which you can see here from showing the predictions on one of these modules here in blue compared to the measurements. But there was actually one qualitative difference between the predictions and the measurements, in that notice that the predictions, even though at about the right place, have a very gradual type of decay at the border, mimicking the behavior of the input transcription factors. But notice that in reality, the measurements of these modules show a very sharp boundary of expression. Okay, so it's been suggested that this ability to convert analog types of inputs into digital outputs is the result of binding cooperativity. Obviously, until this point, we have not incorporated this cooperativity. And so we try to ask if we can make one addition to the model and add cooperativity between transcription factors as they bind DNA, and then see whether we can now perhaps get this type of sharp expression boundary. And so the way we add this cooperativity is between transcription factors only of the same type, and I'll get to this point later. And we add it by giving a bonus to configurations in which there's two molecules of the same type that are bound in proximity to each other, and this bonus decays with the distance between the bound molecules. And so adding this component to the model, we could see that indeed we now, here it is for this one module, and we see it for other modules. We could now recapitulate this behavior that's observed in the measurement by adding this cooperativity. Okay, so that's essentially the model, and now I'll tell you about some validations of this against the system, and then some design principles that we think we were able to obtain by looking at how this model is actually producing the patterns. Okay, so here are the results on the training data. What's shown here is all the different modules. Each module, you see the predictions in blue, you see the measurements in orange, and largely you can see pretty good agreement between the predictions and the measurements, of course, far from perfect. This is for about 80% of the input modules, those that actually worked. Here are these, the 20% which did not work, where the model could not well predict the actual measurements. But the cases where the model did not work are actually, we think, equally interesting, because they in fact tell us about which aspects of the phenomena the model is not yet able to explain, and by that maybe direct us to what's actually missing still from the system. And for example, here you can see that there's something common to cases that we missed. The cases that we missed typically are not able to produce expression in the middle portion of the embryo, suggesting that maybe there's a missing activator in the system, and this is actually one direction that we're now exploring. Okay, but of course these are results on the training data, and what you really would like to see in the ultimate test is results on test data, where you can learn a model from some subset of the modules, and now after you learn the model, you want to play this game where you give the model a sequence that it never saw before, and you ask it to predict where that sequence will give rise to pattern. Okay, so we've done this for two different sets of modules that were measured by others, and actually were measured after we developed our model. Here are the results of then having the model learned on the input modules, and then looking at its predictions in 11 different cis-regulatory modules measured by the group of Steve Small from NYU. And you can see, again, results not far from perfect, but in about the same number of cases that we can predict on the training data, we can actually predict quite well the positions of where these sequences would be expressed, only based on their DNA sequence. Okay, and of course, until now, I've been evaluating these kind of subjectively by using this tick mark type of evaluation. You'd of course like to see a more objective type of evaluation, and so I won't go into the details of how exactly this was done, but you can produce these standard ROC or AUC types of plots, and basically random predictions are on this diagonal line, and the predictions that we can see for the model, again, far from perfect, but much, much better than random, indicating that the system, the model has actually captured some of the essence behind how pattern formation is governed in the system, and it's doing it better than various permuted models in which you permute the various binding specificities of the transcription factors. Okay, so if you buy into these validations, then what's really nice about having a model is that the model is actually telling you the process by which transcription is working in the system, and so you can inspect the model to see what it might be telling you about design principles in the system. And so we looked at a few aspects of this. The first question was, remember that we're going over all possible configurations of factor molecules bound to DNA. So after we learn the model, we can ask, in retrospect, which configurations are more likely, those in which there's many bound molecules on the DNA or those which have a very few number of binding events per configuration. And what we find is that configurations with many binding events, together covering about 20% of the DNA sequence of a given module, corresponding to dozens of binding events, those types of configurations we find are much more likely than these types of configurations. And if you look at it more, you find that about 50% of these binding events are coming from binding to very strong binding sites, as one might expect, but the other 50% is actually coming from binding events that occur to very weak types of binding sites. Okay? And so we think that both strong sites but also weak binding sites are significantly contributing and carry information about how pattern formation is governed in the system. As another validation for this, we also compared the performance of models that are only allowed to use the K top strong binding sites per transcription factor at each module, and you could see performance is improving as you allow more and more use of binding sites per transcription factor. But even when you allow 20 binding sites per factor per module, when you threshold at that level, you're still doing worse than when you allow the full contribution from all the weak types of binding sites.
Question?
Yeah.
So this is posterior inference conditional on gene expression data. You're inferring the probabilities of different configurations of transcription factors based on the expression patterns.
So you're
Derived directly from the model that you specified with the thermodynamic probabilities and concentrations.
Yeah. So initially we use the gene expression data to fit the parameters. But once the parameters are fit, what I'm showing you now is just looking at the model and now using those parameters, asking which configurations of the DNA binding component are found to be more likely. And so here we're not using the expression. We use the expression to fit the parameters, but now we're just looking at what happens when you run this model on the DNA.
Parameters here are what? Concentrations of transcription factors?
So just the one parameter which just scales the level of the concentration. So we know the gradient of expression,
Yep.
and we just need to scale those because those are measured. It's a technical issue of those are measured in relative amounts along the embryo, and so if we don't have the information if, for example, factor A and factor B have in general a scaling factor of 5 between their concentrations. So that's just one parameter for that, and the other is for translating a bound molecule to what its contribution to transcription will be.
How did you calculate the binding energies for the different sites?
Yeah, so those are we basically use the standard approximation of taking PSSMs and converting those into apparent free energies. And yeah, that's an approximation, but a standard one that's all we can do here because that's the input, this binding what we know about the binding specificity for these factors typically comes from a relatively small number of bound sites, so it's hard to do anything more complicated than that. Yeah.
You mentioned that, this is that the number of binding sites is critical if you want. But what about the nature of the factors? Are there any factors which sort of correspond to relatively few binding sites, which could conceivably be eliminated without harming the prediction?
Yeah, so there is variation between, if you look per factor, between how many binding events occur per factor, but largely all of them are really coding the DNA of modules that they recognize. Not all factors work on all modules, of course, but when a factor works on a module, at least by the predictions, it's despite variations, it's still quite a lot of binding events. Okay, so that's contribution of weak sites and many different binding events. And next you can ask, okay, you have a lot of binding events, but you could still imagine two situations. One in which these binding events are kind of organized, and the other in which you have a lot of binding events, but they're kind of random. And again, asking the model which are more likely, you find that configurations that are more organized, and specifically organized in a way that factor molecules of the same type are bound close to each other, are found more likely than just random organizations, and also more than organizations in which you have specific factor A and factor B that are binding in proximity to each other. And so this kind of clustering, this homotypic clustering of sites is suggestive, of course, of binding cooperativity, which, if you recall, at the beginning I mentioned is required in order to generate the sharp expression patterns of the system. Okay, so if you just look at the DNA content of these modules, you find, without any model even, you find that there is tend to be more clustering of these sites per transcription factor within the space of these given modules. Okay, another interesting question you can ask is, okay, we talked about the number of binding events, but what about the actual identity of the factors that are binding? Okay, so the intuition here is that if you look at a given module and you look at a position in which the module is expressed, you expect that the configurations that are dominant will be ones in which there's activator molecules bound. And if you look at positions in which the module is not expressed, you'll see repressor molecules bound. Okay? And largely this is the situation that we saw in the analysis, but what I want to point out is that the situation was really not this clean, but it actually looked more like this, where although you saw enrichment for configurations with more activators at positions where the module is expressed, you also had binding events of repressors at positions where the module is expressed, and conversely binding of activators at positions where the module is not expressed. Okay? So initially this seems like a problem, and maybe a problem with the model, and it might be a problem with the model, but if you think about it, it actually makes a lot of sense integrating what I told you before, that we actually find many different binding sites per factor at a given module, including binding to many weak types of sites. And so if you think about that, it's going to be very hard to design a sequence that not only would have the right inputs to the factors you want, but would also avoid having the inputs to all the factors you don't want, including avoiding these weak types of binding sites. And so what we think is going on is that largely this picture is actually correct, in that the reality of the system is that it works where some of the binding events are kind of individually not what you would want, but by this mass action of enriching for the right types of binding events, that is sufficient for the system to generate the right types of patterns as predicted by the model. Okay. If you now just look at the system, another intriguing question is you can see that from a global view, there's many different patterns that are produced at various different boundaries along the anterior-posterior axis, but these are produced from only 8 different factors that have very well-defined boundaries. So one interesting question is, how can you go from a limited number of boundaries into a much larger space of different boundaries of output? And one kind of interesting particular case that we found was in the way you do this by tuning the DNA sequence, in one case between an activator and a repressor. So one activator in the system is a transcription factor called caudal, another is called hunchback. Their binding specificities are actually very similar. They both have a consensus of T, but there's also differences between these binding preferences. And so what we found, and one more thing, the activator caudal is expressed, the activator caudal is here, it's expressed from the posterior to the anterior, and hunchback is expressed in 2 domains, and one domain also is more a posterior domain. And so what you find is that you can actually get, here are examples of 2 modules that are determined, whose boundary, shown here, is different. And the boundary, according to the predictions of the model, is determined by how you specifically tune the DNA sequence between the preferences, the differential preferences to caudal and hunchback. So the more you have the DNA sequence tuned such that it likes the caudal over the hunchback, the more caudal will be able to compete out a hunchback, since they bind very similar regions in the DNA, and push the boundary towards the anterior end. And the more the sequence favors hunchback, it'll push it more towards the posterior end. So that's one principle by which only by changing the DNA sequence you can control. You can control, even though you're using the same input factors, where exactly you'll put the expression boundary. Okay, and finally, if you you can kind of zoom out and look at how the system is generally organized by computing the asking the model to compute the occupancy of the different activators and repressors along the anterior-posterior axis, and by that you can get an idea of which activators and which repressors are controlling the pattern in each of these different modules. And if you zoom out from this, you find that in general the way the system is organized is that activation occurs by about 1 to 2 different types of activators acting on a given module, with some choice of which activator to choose at the ends of the embryo and at the middle, whereas repression occurs by actually integrating multiple repressor inputs, with coextensive repressors being disfavored, and almost no evidence for direct autorepression in the system. Okay, and we also find that all of these design principles actually give the modules, give the system a fair degree of robustness against mutations, where if you look at the predictions that you have at one cis-regulatory module and you start to make random point mutations and also deletion mutations that are not shown here, then you see that even with a fair bit of different point mutations, you still, on the average, can produce the right expression, about the right expression pattern. And that, we think, arises from the way the system is designed in terms of by the usage, for example, of multiple binding events, including contributions from these weak types of sites. Okay, so let me just summarize this part of the talk. I told you about a thermodynamic model that we developed for transcriptional control. The model doesn't actually have an explicit network structure, but the way that which factors regulate which genes is just determined by modeling the molecular interactions that are going on. And the model explicitly models the binding of factors to DNA, including taking competition between factors into account. And we validate the model by showing that it can do a fairly good job at predicting spatial expression patterns from DNA sequence alone. And then using this model, we could suggest several design principles that are going on in the system. We find that modules are highly occupied by transcription factors, including contributions, important contributions from weak types of binding sites. We find that sites for the same factor, but not for different factors, tend to locally cluster within these modules. And this local clustering is probably there for cooperative interactions between factors, which are in turn needed to generate the sharp patterns of the system. And we find that although the system is enriching for the right types of binding events, you actually have many binding events that you would deem initially inappropriate, but that's the reality of the system, and that is actually still sufficient to enrich for the right types of sites in order to produce the right patterns. And finally, we find that in order to produce the patterns, you integrate inputs from multiple transcription factors in a combinatorial fashion. Okay, so how much time do I have? 10 minutes. Okay. So I told you about this model, which tried to look at this problem from the perspective of modeling the binding specificities and interactions between transcription factors. Obviously, there's many of these different components that I described before that are missing from the description of the model. One major component is the chromatin structure, because as you know, in eukaryotic cells, DNA is not found in its naked form, but actually is highly packaged into chromatin, where the basic repeating unit is a nucleosome around which 147 base pairs of DNA are tightly wrapped. And so why is this important for the process? Well, if you look at the structure of transcription factors, you find that often transcription factors, when they bind DNA, they need to wrap around the site that they recognize. And so these two binding events cannot simultaneously happen. And if you combine that with the fact that about 75 to 90% of all the DNA in every eukaryotic cell is packaged into this nucleosome structure, the immediate conclusion is that the detailed positions of nucleosomes along the genome is going to be very important for understanding transcriptional regulation. And so if that's important, one question you can ask is, what are the factors that determine where nucleosomes will be positioned along DNA sequences? And there's probably many factors. One that we have been exploring is, what is the contribution of the DNA sequence to the positioning of nucleosomes along the genome? And so to look at that, you can do an experiment in which you measure the locations of nucleosomes along the genome. And it turns out if you extract these sequences and you align them, just take these sequences and center align them, you find a very striking pattern of specific dinucleotides showing periodicity along the length of the nucleosome that's actually been observed over 20 years ago. Similar periodicities were observed when extracting nucleosome sequences from chicken. You find these periodicities for these specific dinucleotides, and also superimposed with them or out of phase with them are periodicities for other dinucleotides. What's nice specifically about these ones that you find is that these specific dinucleotides are known to have a role in the ability of DNA to sharply bend, which is the properties that you would imagine would be important for the sequence specificity of the nucleosome, because the DNA is sharply bent around the histone octamer in the nucleosome structure. Okay, so and if you allow yourself to kind of realign these sequences a little bit, you can see the pattern much more clearly. And so based on these, we suggested a statistical model that describes the DNA sequence preferences of nucleosomes, and we could show that using this statistical model, you could predict the positions of nucleosomes in genomes with an accuracy that's, again, far from perfect, but much better than random. And in recent years, there's been, with additional data and improved types of models, others, and we have now work in progress showing that you can actually push these accuracies much further. All of these suggesting that at least one big component of determining where nucleosomes are positioned along the genome is determined by the DNA sequence itself. We've now collected nucleosome sequences from multiple species and did some measurements in also in fly and human, and in all the cases you find these striking periodicities appearing in collections of nucleosomes from all these species. Okay, so so if you think of nucleosome positions somewhat being partially determined by the genomic sequence itself, the next obvious question is then what functional roles might this encoding have? How might the genome be utilizing these nucleosome positioning codes for various of its functions? And so that's the next big questions that we're working on in this field. I'll just mention, but skip over this for lack of time, that now we've been using the amazing power of high throughput sequencing to measure, instead of 200 nucleosome sequences that we've been working with before, about 500,000 nucleosome sequences. The data is really quite amazing. There's no analysis that you have to do. You just map it to the genome. You find where nucleosomes are. There's no comparison to trying to look at this from DNA microarrays, and this type of data is really what's enabling now much better ability to discover properties to work in this area. And so let me just skip over this, but just show you that if you now look at the predictive power of or an accuracy after you construct your model from a lot more data, then you can see that this is in predictions in the yeast genome. Your ability to predict nucleosome positions is becoming quite good. In fact, if you look at nucleosomes that are very stable, your predictive power is almost perfect in predicting these nucleosomes. from and separating these ones from what are linker DNA sequences, regions that are not bound by nucleosomes. Okay, but so I just want to end then by showing two properties in which the yeast genome is utilizing these positioning signals for various of its functions. One is we had this hypothesis that maybe the genome is utilizing these sequences to enhance the accessibility of transcription factors or to help it in solving the problem of how to direct transcription factors to their appropriate sites in the genome. Okay, and this is a, if you think about it, it's a quite challenging problem that the cell has because the binding specificity of transcription factors is often very small. Sometimes the recognition site consists of 5 or 6 different nucleotides, and so you find these sequences all over the genome, but somehow the factors know how to go to the functional sites and not to all the happenstance sites that occur in the genome. And so one hypothesis that we had is perhaps the functional sites are arranged in such a way that they tend to reside in regions that are largely free of nucleosomes, whereas the other sites, the ones that are non-functional, just happen to sit on the genome average, which on average would place them under a nucleosome. Okay, and so to look at that, we took the predictions of nucleosome positions that our model generates on the yeast genome, and now we took a database of binding sites for transcription factors, functional and non-functional, where a functional site had to be bound according to chip-chip experiments done by Rick Young's lab, and also the site had to be conserved in at least 4 different yeast species. And then the non-functional sites are the kind of the consensus sites for the factors, but those that don't have either one of these two properties. And so if you look at that and you look at the predictions of the model of nucleosome positions, which of course did not incorporate any information on binding specificity, then you find that functional sites for most transcription factors and for about 40% in a statistically significant way tend to reside more in regions that are predicted by the model to be devoid of nucleosomes compared to regions that have a higher nucleosome occupancy. This is also consistent with experimental results of where nucleosomes are actually positioned in vivo, and work from Alex Hartming's lab also showed that you can incorporate this information on nucleosome positions to improve your ability to discover motifs by basically helping you in the signal-to-noise ratio by taking out sequences that are more likely to be bound by nucleosomes and therefore don't participate in the search space of transcription factors. And so we think that at least one function of this nucleosome positioning signals is to help transcription factors in binding to their appropriate sites in the genome. Okay, and the final result is regarding transcription initiation. You can look at the predictions across all the different genes if you align all the genes according to their translation or transcription start site. If you do that, you find that the predictions have a very strong peak right around the start of translation, or about 40 base pairs upstream of that. If you superimpose on that now locations of Tata boxes, you find that Tata boxes tend to appear right in this linker DNA region in between 2 neighboring nucleosomes, suggesting that on average, the yeast genome is encoding a nucleosome organization that's facilitating transcription initiation by positioning the nucleosomes such that they free up the Tata box for binding of the transcription initiation machinery. And again, this very strong nucleosome sitting right on the translation start site is also seen in experimental measurements of nucleosomes in vivo, and in fact also in various different organisms, including human, where a nucleosome map was recently made available. So to summarize this part, I told you about a model that we were developing for trying to understand where nucleosomes are positioned along genomes from properties of the DNA sequence, and then applying this model to the genome, we suggest that the yeast genome at least is utilizing these signals for various functions, including enhancing the accessibility of transcription factors to their right sites in the genome, and also for facilitating transcription initiation by freeing up the locations where the initiation machinery needs to bind. Okay, so going back to this picture that I started with, obviously we're very far from this understanding of how DNA sequences encode for particular behaviors. There's many of these different components that we have not integrated at all, and some that, and for the others, we still don't really understand how they function. But and so in general, and just to show you that we also don't take ourselves too seriously, we definitely agree with a famous statement by the statistician George Box, who said that all models are wrong, but some are useful. But hopefully what I convince you in this talk is that if you choose the appropriate experimental system and model, and then by developing this approach of developing quantitative models, we can actually get useful insights into the workings of these systems. And so with that, I'd just like to acknowledge the people that did this work. This is our group at Weizmann. The work on the transcriptional networks in Fly was done by Tali Tzadka, a PhD student in the group. The work on the nucleosome is done by Yair Field and Noam Kaplan, two PhD students in the group, together with Eilon Sharon and Yaniv Lubling. The work on the Fly is in collaboration with the lab of Ulrika Gall from Rockefeller University, and the nucleosome projects are in collaboration with the lab of Jonathan Widom from Northwestern University. And with that, I'd like to thank you for your attention.
Eran, so in your first page cartoon, I saw you say that you mentioned the orientation, but in your model, did you find the orientation really play a significant role to predict the gene expression pattern?
So we did not include orientation. We did not model orientation in the model.
Oh.
So basically, for us, the activity of the factor was equally active if it was bound in each orientation, and obviously that's among one of the components that we still have not integrated into the description of the model.
I have a quick question. So in the case of Drosophila, which species were you looking at, and have you checked how your model predicts things in related species, now that some are available, and whether the quality of prediction degrades with distance, or not?
Yeah, so first of all, the models we were training on were all done in Drosophila melanogaster, but one of the validations that I kind of skipped over was an interesting validation on the ability to predict these activity of the modules in pseudo-obscura. And in pseudo-obscura, our collaborators have actually done measurements of the activity patterns of some of these modules. And the intriguing experimental observation is that you find that orthologous modules have huge sequence divergence. About 50% of the DNA sequence, if you align and count bases that change, about 50% has actually changed. But remarkably, when they take these sequences, they put them in Drosophila melanogaster. Despite this sequence divergence, they produce nearly the identical expression patterns. And so
That's what I'm asking.
yeah, and so those are recent, very intriguing experimental observations. And one of the validations that I showed is that actually the model, again, far from perfect, but the model is actually able to predict these the same expression patterns for these orthologous modules, despite the sequence divergence. Yeah.
I have a question to your comparativity parameters. Why do you only take the two into account the same transcription factors and not two different ones? I didn't quite get it.
Yeah. So there was two reasons for that. One is simplicity. So if you have only a parameter for cooperativity between the same factor, that's only 8 parameters as opposed to 8 squared parameters if you model all pairwise cooperative interactions. But this was also motivated by analysis of the DNA sequence, whereby you find that the signal is that binding sites for the same factor tend to locally cluster within the modules, but not binding sites for different types of factors. So that was suggestive that also the cooperative interactions, or the dominant ones that are going on, are between factors of the same type. And largely that may be true. We know that it's not true. We know of several specific examples. where one of the factors bicoid, another one hunchback. Specifically, in one or two of these modules, there's been documented cooperative interactions between them. So obviously, that's something that that we don't model, and in fact, those modules are ones that the model cannot explain also.
Going back to that question about orientation, when you see the co-clustering of the binding sites, do those tend to be on the same strand? Because that might be an indication of whether that's...
Yeah, and we haven't looked at that. It's an interesting question. Martha?
Eran, if you look at multi-species sequence alignments, and then you look at where you predict the nucleosomes to be positioned, in the cases where they're not aligned, do you see any favored offsets? So is there a preferred periodicity to how far a nucleosome will shift?
Yeah, we're starting to look, the whole evolutionary aspect of this is very interesting, and we're starting to look at that. I don't know the answer to this question. In general, it's a complicated aspect to look at, because unlike transcription factors where the signal is very localized to short words, and you can use sequence alignment to know whether a specific site is still functional or not, largely speaking, with the nucleosome, all 140, your signal is integrated pretty weakly per position, but over 147 bases. And so sequence alignment, for example, is not very useful for looking at these types of questions, because you can have changes in one region that are easily compensated by changes 100 bases from it. And so we're, yeah.
I'm not sure my question really came across then.
Okay.
So if you separately predict where the nucleosomes will be positioned in each of the genomes,
Mm-hmm.
and then say, okay, these guys are aligned, these guys look like they're offset.
Yeah, okay. So we're starting to look at that problem. Very preliminary results that we have show that we do find changes in the nucleosome organization at some promoters, not in others. And some of these actually might be interestingly associated with different changes in the function of the genes between the different species. But this is a direction we're just starting to look at.
Right at the beginning, you started off by calculating thermodynamic energies and interpreting those as probabilities. Are you able to calculate the partition function?
Yeah, so that's exactly, so the dynamic programming, that's the whole trick behind how this is working. Turns out you can do dynamic programming where you can compute the partition function, and that's what allows you to, under the thermodynamic equilibrium assumption, compute exactly the probability of the system being in each of one of these states.
In the enhancer model, did you ever try shuffling the columns of the PWMs right at the beginning and then fitting to see how much it mattered what PWMs were used?
Yeah, so that was some of the, in those cross-validation curves, some of these permutations that we were doing were exactly doing permutations on the columns of the binding specificities of the factors. And yeah, and those, if you fit those, they have significantly reduced performance from the real data.
Okay, thanks very much.
Thank you.